You're listening to Don't Waste Water.
People of my team say, "You are totally crazy. It's not possible." I say, why?
Hello, bonjour, and welcome to the Don't Waste Water podcast.
We have been evaluated by Beijing, Chinese CDC, and they made a publication. The conclusion is that mica deliver more accurate residue compared to the ISO method.
I'm your host, Antoine Walter, and in today's episode, I'm delighted to welcome Sam Ducamp as my guest.
Take 10 liters of liquid, put Legionella on it, mix it, and split it to 10 different laboratories. And inside the bottle, it was 10 to the 6 Legionella pneumophila per liter. Not few, 10 to the 6. And we got laboratory that obtained 0. Accredited laboratory that obtained 0.
Sam is the CEO and founder of Diamidex.
With our solution, he saved every year 75,000 euros because
Diamidex helps labs with improved productivity by implementing rapid, reliable, and customer-focused detection methods.
it will take 2 years and around 2 million euros. And we have the 2 years if we are alive, but we have not the 2 million euros.
And before we start, let me thank my sponsors, Simple Lab and Bluefoot Membranes.
You have a dead body in front of you and you ask him a question. Do you think that he will answer?
I hope not. When is the last time you got offered to invest in a private company? If you're like me, that's probably never. I mean, I've crowdfunded one of my friend's music album and even one of my other friends' continued studies. But never did I come right in time to participate in a water company's crowdfunding efforts. And that's even more surprising when you think that two of my podcast alumni have actually been crowdfunding in the past 2 years, Can-Do and Salinity Solutions, but I missed those boats, and I have to say, when I see how they've both developed, I'm pissed I did miss them. So here's where I need to tell you, this is not financial advice. Always consult with qualified financial professionals before making investment decisions, and I'm not responsible for any actions you take based on the information discussed. But today, you'll hear the full drill through Diamidex as a company, but also as an investment vehicle, currently crowdfunding, and this until the 25th of June. Regardless of whether you invest or not, I would encourage you to have a look at the crowdfunding page, because I believe it's a very rare glimpse behind the scenes of a water company's cap table. Sure, you'll get key information on Diamidex, like the market they serve, their business model, the price of their equipment and consumables, or the number of tests they already performed, but you'll also get their detailed valuation, who owns what percentage of the company, and for instance, how much equity Pure Ventures got for their 2 million in the 2.5 million funding round they led in November 2023. You'll also get to know what Diamidex intends to do with the funds they raise, depending on how much they gather, and receive a snapshot of the exits they foresee down the line. Nothing revolutionary on that front, though you'll get more crispy details in today's interview. So here's the lens I propose you to wear during today's episode. Should you invest in Diamidex? Is it an opportunity that's too good to be missed, or should you sit a bit longer on the fence? You'll see that I'm also applying those questions to myself. Happy to discuss with you down the line how convinced you'll be by Sam's answers. But in a minute, you'll hear how a 10-year journey from CNRS research director to water tech entrepreneur led to a breakthrough that counts microorganisms 4 to 7 times faster than century-old Petri dish methods, why this technology delivers results in 6 hours for E. coli instead of 24 hours, and 2 days for Legionella instead of 10 days, while maintaining exact accuracy, if not simply improving it. What's the secret sauce combining optics, click chemistry, which won the Nobel Prize in 2022, and machine learning that makes invisible microbes visible and countable at the microcolony stage? How major pharmaceutical and beverage companies are knocking on Diamidex's door, with one global partnership launching end of last year and acquisition talks already underway, and of course, getting to the bone of their razor blade business model targeting 75 to 8% recurring revenue from consumables, with equipment potentially given for free to high-volume customers generating 500k annually. This and much more, right after I remind you that if you like what you hear, please take this episode and share it with a friend, a colleague, your boss, or your team, and I'll meet you on the other side. Hi, Sam.
Hello.
Welcome to the show.
Nice to meet you again.
I'm pumped up to have you today because, as you say, we meet again. I've seen you pitching to investors, but I want to start with a bang. And actually, you're in a very specific field. You're counting microorganisms, which, if I say it like that, sounds like dry, but there's a lot to it. And counting microorganisms, when I was in my water engineering school, is something we did by pouring water in a Petri dish, letting it grow for a while, then coming back the week after roughly, and then counting. And I thought that's an exercise which you do because you're in engineering school. And then it turned out that is simply the way it's done. Like in the 21st century, we still do it like that. And that is where you come in and you say you have a better solution. So what do you enable?
By training, I'm a microbiologist, and I made exactly the same conclusion. It's crazy to say that we are in 2025, and in all the laboratory all around the world, technicians take a membrane, put water and filter the water in the membrane for 100 milliliter, 200 milliliter, and after they put the membrane on the plate and wait for days on specific media to be able to enumerate the correct microbes of interest. Now we are in 2025. What will be the best way to do it in order to obtain exactly the same result as the standard method? Why we are obliged to wait to obtain a colony visible by eye? It's just because microbiologists love to see what they are doing. But in fact, we could see the microbe at the stage of microcolony, which means the culturable cells, the ones that are really important, start to divide and generate a microcolony. But if you have a microcolony, you cannot identify it by eye with a microscope if it's Legionella pneumophila or if it's E. coli or if it's Pseudomonas aeruginosa. So you need to combine an equipment that will allow to detect the microcolony, but also an approach that will label specifically the microbe of interest. And this is not enough because you have a lot of artifacts. So you need to add on top of it machine learning and artificial intelligence to combine and to be able to make the difference between all these objects. And from that, we got the idea to develop a microcolony counter that combine optics, mechatronics, some specific props that are able to label the microbe of interest, and the machine learning. And it was a starting point, of the idea of MICA for microcolony counter.
How fast is it?
For E. coli, we are able to deliver results in 6 hours instead of 24 hours. For Legionella pneumophila, it takes normally 10 days to obtain the result, and we are able to deliver results in 2 days. But the beauty is that we deliver exactly the same result as the one that will be obtained with the standard method. You have a lot of issue with all the other alternative methods that are not able to make the difference between dead, culturable, viable, but non-culturable cells. But with our solution, we are able to detect only the microcolony. So the same that are detected by the plate.
So what you're saying here, and that's something I really want to drill down in a minute, is that you're not the only one who offers a technology which enables to go faster, but you're the only one with a technology that goes faster and is using the same methodology than what microbiologists are doing today, which means it's not disrupting the principle, it's just making the same thing 4 to 7 times faster. I just want to raise the stakes because as we're speaking, you are fundraising. And usually when people are fundraising, first, they don't say it. And second, they do it in very private rooms with specific investors. You went a different route because your funding is open on a crowdfunding platform. The link is in the description if people want to have a look at that. I have the platform open in front of my eyes and I'm sitting on the edge. I don't know if I should be investing. So at the end of our conversation, I guess I will know if I should be investing or not. On my personal benchmark, crowdfunding is a very positive sign of health for your company because that shows that you're trusting it enough to go and make it public. Right now, if you have to give me one argument for me to click on that Invest Now button, what would it be?
There are some competitors that have developed tools that are able to count microcolony also. But the big advantage that we have compared to all these competitors and the one that detect microcolony is that we have designed a tool that could be used for all the microbes. Usually you have startups that are focused on, let's say, the total viable count. So they are able only to detect all the microbes present in the sample, but they are not able to detect E. coli or to detect Pseudomonas aeruginosa or whatever are important to be detected. With the technology that we have developed, today we have Pseudomonas aeruginosa, Legionella pneumophila, Escherichia coli, but tomorrow we'll have coliform, we could have Enterobacteria. We have no limitation. This is the beauty of what we have done, because the fact that we combine the equipment that is able to scan the membrane and the dedicated probe that labels specifically the microbes and the machine learning could be applied for all the microbes. You just need to have the right probe. And this, we have the capability to do it. With MICA, you have the future gold standard of the microbiology, at least in water. And this is unique because we answer all the need of a laboratory that is performing microbiological analysis for water. Not only one, all of them. And this is totally unique.
We'll explore that. To get to understand how you came up with that, I'd like to just rewind your path. And you've started that entrepreneurial journey, if I'm right, roughly 10 years ago, at a time where you were a CNRS research director. For people that are not familiar with the French system, CNRS is like the most highly ranked and viewed public research organism in France. And you become mad and you turn into entrepreneurship. What's the story there?
I start my career in the CNRS in 1998. Since the beginning, my goal was to open a company. If I enter in the CNRS, I was saying, okay, I want to open a company, but I need to have a smart ID. And this is not coming just because you ask, oh, I need to have a smart ID, and boom, you have the smart ID. During all my career, I was working with BASF, with Suez, with a lot of different companies that came to me and asked me, look, we have an issue here. We don't understand what is going on, but we need to have the view of fundamental research. One time every 2 years, I say, wow, I have an idea and maybe I could build a company, And I knocked to the door to one of my friends, Jean-François Bloch, is in charge of IP. And I say, look here, Jean-François, I have this idea. What do you think? And he said, no interest. Okay, I will try again, And 2, 3, 4 years after, I repeat. And in 2010, with one of my new fancy discovery, with my friend Boris Bozel from the chemist laboratory, I explained to Jean-François, look, here is a new way to detect microbes. And that time he said, wow, I think that you have something. And I was very lucky because just 2 years after, in 2012, in France, you got the opportunity to be supported to develop new technology from the SATT, which are some public companies that are able to support the development of innovation in order to build company. So from 2012 to 2014, I have been incubated in the CNRS in order to develop this technology. And at the end of 2014, I say, okay, now I have enough to say, okay, I will start to open a company. And I decided to build the company at that time. So end of 2014. And the technology was based on the click chemistry. We have developed the capability to label specifically a microbes based on the incorporation of one molecule that was metabolized only by one microbe. And by doing so, we were able to detect only Legionella pneumophila. But it was just the idea, In 2015, I was very lucky because Alstom Power knocked to my door and say, wow, we have heard that you have a new tool that is able to label specifically Legionella pneumophila. And we know that we have issue with cooling tower and we need to have tools that will be able to enumerate earlier these microbes. So we were able to develop a solution. And this took a lot of years.
Just to be clear, and for the muggle and the layman, which I am here, when you say a tool, that means that you give something to eat to all the bacteria, and it happens that only Legionella is developing a kind of reaction to what they eat, so that when then you look for the tracer, it will only appear in that specific microbial species and not in all the others. And so that was your differentiated innovation initially.
Initially, yes. So just to make it very simple, it's like you fish, you will be able to catch only Legionella because the molecule that we have developed is metabolized by Legionella pneumophila, incorporated in the macromolecule, and after on the surface, you have some anchor on the surface. And by click chemistry, that obtained the Nobel Prize in 2022, you are able to attach a fluorescence probe. So you make invisible visible faster.
That means that at that stage, you have found a way to put a tracer on Legionella pneumophilia, and you have Alstom knocking at your door and saying, by the way, we would love to detect Legionella,
Exactly.
and you seem to have something. But you still need to develop all the in-between steps, which
Exactly.
is having something to watch it, to measure it. How does that come into music? How long does it take?
It's a long journey, because you have the idea, you have the probe, but you have not the solution. So you say, okay, now that I have the probe, I need to understand what is needed by the customer and what is the standard rule. And the rule is the Petri dish. And what the customer wants is colony forming unit. It's what is the number of culturable microbes. So we say, okay, we could apply this probe in between the Petri dish plate and the membrane. This probe will be metabolized, so the microcolony will be generated. And if we have a reader that will be able to read the fluorescence that will be generated by the microcolony, it will be possible after by image recognition and artificial intelligence to detect and enumerate the microbes. So it took us 7 years to develop the solution. But we developed not only the solution for Legionella pneumophila, we developed the concept because the probe could be something else. It could be an enzyme activity, it could be antibodies, it could be fluorescence in situ hybridation. The only thing is that at the end of the day, when you have the microcolony of interest need to be fluorescent to be detected. Whatever is the way.
Let's hear from my sponsor, Simple Lab. Testing water and everything around it should be simple. With Simple Lab, you choose what to test, from drinking water and wastewater to soil, air, food, and surfaces, and they handle the rest. Kits arrive at your door with free round-trip shipping, samples go to certified labs, and results come back fast, concise, and actionable. Trusted by leading brands and growing teams, Simple Lab offers hundreds of ways to better understand your environment. Get started today at gosimplelab.com. It's the first link in the description. Thanks Simple Lab for supporting my work, and back to me. Something I'd like to understand here is that you mentioned how you're a microbiologist. You also explained how your co-founder is a chemist. And now you mentioned that you need optics and that you need artificial intelligence and machine learning. Did you teach all of that to yourself by evening classes, or did you get some external support?
It was very funny because when I say, hey, look, my team, we will start to make artificial intelligence and optics and mechatronics. People of my team say, you are totally crazy. It's not possible. I say, why? We just have to attract the right people in the company. And we were very lucky because we attract a really smart person on optics. We attract smart person on machine learning. And all together, we build the solution. Me, I was just able to have the vision, but the vision is not enough. You need to have a team of experts. And it's what I have built. Usually, you have a lot of turnover. But here in the company that we have built together, we have people that stay for life. It's a family.
But from an entrepreneurial perspective, like you have the golden goose, you have a process which requires a recurrent revenue because it is a Petri dish with modified stuff inside. You have the patented chemical hook. So you could be just churning IP and recurring revenue. And you, instead of doing that, you say, no, I want to do what's the absolute hardest everybody agrees on in entrepreneurial endeavors, which is I want to build hardware.
Yes.
Why did you pick that? Why not just the recurring and let the recurring run and partner with someone to do the hardware? Why did you want to have everything under one roof?
Because everything is linked. If you are not able to produce the equipment, you are not able to capture the image. If you are not able to capture the image, you are not able to generate the artificial intelligence. And if you have a supplier that starts to modify your equipment, the machine learning that will be developed will not be able to detect the microbe because the equipment will be modified. So it's a synergy in between everything together. The solution starts with the equipment. When the equipment is finished, we could start to identify the right probe and the right way to detect the microbes by machine learning. So we need first equipment. Without the equipment, we cannot do it.
So that means the first step in the ladder was to produce your own equipment,
Exactly.
which, in terms of timeline, when did you have the first
Prototype.
which was worth of that name?
So we got the first prototype, I think it was 2019, and we got the first prototype of the solution, end of 2021. And we start to sell this first prototype solution to customer, and we got a lot of feedback from them and say, look, it's nice, but you need to stick in front of the equipment and you need to load cassette by cassette, one by one. And this is not possible because the technician cannot stay in front of the machine. And wow, your cost per sample is really expensive. So it's nice for emergency sample, but if we want to perform high number of analysis, it's too expensive. So during 2 years, we collect information and we develop a new solution that we call Mica Advance. And this Mica Advance, we believe that today answer all the need of the customer because one equipment is able to perform 500 scan, 500 membrane analysis per day, which is huge. You need just 1 minute per scan, and in 1 minute you obtain the result. After the incubation and after the process, you just put the membrane in the machine and the machine read, and 1 minute after you have the result. We release final equipment only in 2024. So it's very recent.
Couple of questions on that one. You say 500 per day. I guess your customers must be laboratories, right?
That's correct.
How many analyses do they typically run in a day? Like with one equipment?
One equipment is enough today. We discuss with laboratories that perform 300 to 500 analyses per day, which means that one equipment is enough for them.
So you're covering all the daily needs.
Yes, exactly.
My second question is on the timeline. You mentioned how your final Mica device was ready in 2024. Do you produce it yourself?
Yes.
Okay, so everything from the screws to the software is all in-house.
Yes.
Where do you produce?
In Gardanne, close to Marseille. The beauty also of the equipment is that we use only spare parts that are present in the market. Everything, the camera, the LEDs, all the electronics are spare parts that are available. It's very simple. It's like Lego. You plug and that's it. We want to deliver simple solution. And I'm very happy because when I install an equipment in a laboratory in Macau, you say, wow, after just 1 day, I have been trained. It's finished. And now I'm able to double my productivity with your equipment. And when a new technician came in the laboratory, you just give him a sheet of paper with the explanation, and it was trained like that, boom. When you have a laboratory that want to perform Legionella pneumophila in the technician need to be trained during 6 months to be able to do the job correctly. Here, in 1 day, you obtain the result. A technician need between 20 minutes and 40 minutes per sample to perform the analysis. With our solution, you need only 4 minutes. So you see the increase in productivity.
5 to 10 times faster on the analysis itself. The other thing with visual inspection with a human is that a human is subjective. Like a human will count a certain way, another human will count a different way, whereas a machine is counting
Always
without any sentiment in it.
So what I'm saying always is, if the machine make a mistake, the machine will make always the same mistake. But at least you will be reproducible. AFNOR requests me to make a publication on it.
We just need to explain. AFNOR is the French Norm Institute.
Yes, thank you.
Sorry, didn't want to cut you off.
I send 10 times, I take 10 liters of liquid, put Legionella on it, mix it, and split it to 10 different laboratories. And inside the bottle, it was 10 to the 6 Legionella pneumophila per liter. Not few, 10 to the 6. And we got laboratory that obtain 0. Accredited laboratory that obtain 0. So it's crazy. And you got 2 orders of magnitude of variability in between the laboratory. When we done exactly the same with our equipment, we got plus or minus 20%. So the beauty is clearly demonstrated, because you have a solution that is very simple to be used. You don't need to have a skilled people. You have result after 2 days instead of 2 weeks. And you just need 4 minutes instead of 20 minutes. And the human error is disappearing.
I have to be the devil's advocate. It's more expensive because there is a capex barrier to that, which is they need to first buy an equipment when a Petri dish arguably is an inexpensive piece of glass.
I agree with you, but I will give you it's another microbes. So it's not Legionella pneumophila, it's Lactobacillus, which is a spoilage bacteria for fruit juice. So it's not exactly the same market, but just to give you an idea, and I will go back after to Legionella. We made a webinar, and one of our customers in Greece say, and we have not paid him to say it, so I was very wow. With our solution, he saved every year 75,000 euros because he was able to deliver result in 1 day instead of 9 days. He was able to reduce the time in between the production batch. He was able to release product earlier. So he saved a lot of money. Of course, you have to pay the consumable. You have to pay equipment. But when a laboratory with one technician could perform 200 analyses where before he was able to perform 100, so he doubled the number of tests what he could do, and when he invoiced after 2 days, instead of invoicing after 2 weeks, he reduced the eating of the plate because he used only one plate and for only 2 days, where before he was using 5 plates for 10 days. Of course, we are more expensive, but not so much. And your customer now knock to the door to the lab in Macau, say, I prefer to use your solution and pay a little bit more in order to have result in 2 days because I'm more safe when you control the risk.
Have you ever heard of a membrane that refuses to break? It sounds uncommon, but it's true. Meet Bluefoot Membranes, a Belgian company that has developed the first and only unbreakable membrane specifically designed for MBR. If you're new here, membrane bioreactor applications. This technology is setting a new standard in wastewater treatment. Water engineers and industry experts have tested this membrane, and no matter how much they pull and twist, the membrane holds strong. Now, it's not just about membranes, it's also about an award-winning and passionate team that doesn't take impossible as an answer. The people at Bluefoot Membranes are driven by a mission to make wastewater reuse the new standard, united by a shared purpose to leave a blue footprint on the world. So yes, they manufacture cool membranes, but they also push the boundaries of what's possible in MBR technology. To learn more about them, check the link to their website in the description. A big thanks to Bluefoot Membranes for sponsoring this episode, and back to me. Your story of the double-blind studies where you sent Legionella-spiked samples to laboratory gives a good hint at what the risk is. Because if you're measuring Legionella and you find a 0 when it's not a 0, then it's going to develop. And if it's going to develop, then you might have people getting sick. And I think I did a full episode on what's the cost of a human life. And I know it's a terrible question to ask, but when you do risk management, that is part of the equation. And when you put that in the equation, even if you put a very low probability that an accredited lab gets a 0, I mean, clearly it happens. But before I go to the regulation aspect of it, you mentioned that you have this Mica Advanced box, and you also mentioned that you have these different parameters which you can measure. Would they all be measured with the same box, or do you need to have like a specific tool for each of them?
So we have the same equipment called Mica Advanced, and after you buy different type of consumable. When we want to detect specifically one microbe, it's based on fluorescence and it's with Mica Advanced. And we have developed another technology called Mica Highlight, which is for the total count. We are able, just by putting light on a membrane in a certain way, and it's a new patented technology, we are able to count the microcolony of all the microbes present, yeast, mold, and bacteria. Just by light. It's very funny. You put light and the microbes become shiny.
But you still need to have like a counting device,
yes,
right?
You need to have equipment and the machine learning system.
Would it be like a light version of the Mica Advance, like a Mica Bitless Advance? Terrible name, don't use that for your marketing.
No. The idea is, for Mica Advance, you have the probe, the machine learning, and the scanning process. For Mica Highlight, you have the machine learning, the scanning process, but instead of having a probe, you have a new way to illuminate the membrane.
I see. You mentioned the various parameters. Do you have a bestseller among them?
I mean, we release Legionella pneumophila first, so it's our bestseller. We'll release E. coli in June, this month. So maybe in 2 years, the bestseller will become E. coli. I don't know. And we released just 1 month ago the total viable count. So we just starting for the new parameter. So today it's Legionella pneumophila, but maybe it will change in the future.
But what do you believe is the most in-demand parameter, which you may have or may not have yet, but is a game changer? I'm wondering if you have an idea of where it might be heading and what would be like the biological parameter or maybe even non-biological parameter.
I will not be able to disclose, this because
Okay.
it was very funny. I was at Aquatech. I present for the first time Mica highlight for the total viable count. And I got some very strange requests. They say, but are you able to count something that is not a microcolony? And I ask, why? Because we have issue here and here. And apparently your technology might be able to do it. I don't know. I have never think about it. The day after, I call my team and I say, look, we might need to check if it's possible. And we got very good result. So I will not say more, but we never know indeed. Maybe it will be something totally different in the future. And maybe it will not be microbiology. But what I can say is when I was in London, I discussed with a lot of laboratory and all of them say, we have a big issue with Mycobacterium. It takes 3 to 4 weeks to obtain the result. And we have people that die every year due to Mycobacterium, and not only tuberculosis. And if you are able to use your technology and reduce the time by 4, it will be fantastic. So we made some proof of concept that we are able to do it, but we have not the money to develop the solution because it's always the same. We need money. And to develop a new solution, based on Mica Advanced, because it would be the same equipment, it will take 2 years and around 2 million euros. And we have the 2 years if we are alive, but we have not the 2 million euros. I don't know, maybe somebody in the audience will have the capability to invest in the company and to support us in the future. Or maybe you have a laboratory that will be interested to have an exclusivity and will invest the 2 million just for one microbiological parameter. Because we have no limit in our technology. So if somebody wants to detect Neisseria fowleri, which is a amoeba that eats the brain, we are able to do it.
So that means you're building a platform,
Exactly.
and now it's about what to add to the platform. And of course, there is an investment linked to that,
And
which
the
is that
revenue.
you can, but the investment coming before the revenue,
Yes.
you have to have a way to finance the flywheel. There is an elephant in the room. I want to address it. The elephant in the room, we touched on it slightly, is the regulation. And the regulation is what comes in the way of most of the approaches which are a bit more clever with lab analyzers. Just to take an example, I looked into the reference for Legionella because I was curious to see when that was put in place. And the test which is used today was implemented in 1998 and revised in 2017, but the revision actually is very cosmetic. So it's really the 1998 test which is still in place. And it is defining how you should do it with exactly what I said in the introduction, what I learned in engineering school, where you clean your sample, you put in the Petri dish and you count it. So that is what regulation is enforcing, which, and that's the problem most of the time, also means that whatever clever approach you find, whatever very cool way you find to be faster, you still have to do your Petri dish and counting.
You are totally right.
Is that the case with your technology as well?
Yes, but because we are enumerating exactly the same as a standard method with exactly the same Petri dish, and we are just looking at the stage of microcolony compared to colony, you have some countries that are more open to use it in replacement of the standard method. Of course, it's something that takes time. It's not something that you could do it in 6 months or 1 year. But in some country, if you use a method that is based on culture, where you are able to count colony forming unit, and where you are using acid shock, when you are using a way to identify Legionella pneumophila, you could use it instead of the standard method. Like I say, we start with Mica Advanced in 2024. Today, we have already 3 laboratories, I was trying to count, that are ISO 17025. 0.025 for Legionella pneumophila. So which means that they present result, they compare the result with the standard method and they obtain the same result. Sometimes it's enough to replace the standard method. Sometimes it's not enough. So after you need to discuss with the Ministry of Health in order to change the regulation. And this will take time. So today, our main customers are using our solution for what we say auto control, not for the mandatory analysis. Let's say we have a customer that must perform an analysis every month for the mandatory analysis. And if he reach a certain threshold, he has the obligation to close and to clean. And close the cooling tower costs a lot of money. So what he do, he tests first with mica, obtain the result after 2 days. And if the result is good, fine, he will make his standard regulatory analysis. And if the result is not good, he will clean before and after perform the standard method and will be always good.
It's interesting because if you go to a Chinese industrial plant, they have these kind of approaches in place for 10, 15 years, roughly since the Chinese government took some very harsh water and wastewater regulations where they effectively closed factories because they found that in terms of economics for the country, it was better to have clean water and clean wastewater. And so you have a lot of these plants where you have the government analyzer. And before the government analyzer, the industrial will have his own sets of analyzers and samplers and ways to collect probes and to measure much more often than the government analyzer because they cannot afford that the government analyzer is the one detecting something because that would mean they're in big trouble. So it's that same approach where you don't wait for the monthly probe to come back and to provide you the bad news. You want to be the owner of your destiny.
Exactly. And the best way to replace the standard method for us is to start to have enough laboratory that will be 17025 ISO, and to have enough academic publication that show that we have equivalent results compared to the standard method. And with all these documents, we will be able to replace the standard method. You talk about China. We have been evaluated by Beijing, Chinese CDC, and they made a publication in Chinese, but the abstract is in English. And in the abstract, the conclusion is that mica deliver more accurate results compared to the ISO method. So it's one publication, but when we will have 100 publications explaining the same, regulation will change. It takes time, and we need to have the time to do it.
That's the thing. I've seen one of your publications, which was showing that you have statistical equivalence in much shorter time. And I'm wondering how many of these publications, you say hundreds, but is there like a track record which shows that if you have so and so much publications, then you wouldn't have to replace the standard. You would have to be accepted by the standard. Like if people want to keep doing their Petri dish stuff, they should be absolutely free to do so. But your technology should be like second line.
It will be state dependent. Let's say today in Canada, we are working on it. I cannot disclose a lot because if my competitor will, but we are working on it. In the Emirates, we are working on it. In China, we are working on it. In different countries in Europe, we are working on it.
You mentioned your competitors, and I do know that usually entrepreneurs don't like to talk about their competitors because you're all in the same market and you don't want to say anything negative. So it's not about saying negative about the others. It's about if I have personally to make an informed bet, I see your approach having better chances to getting regulated one day because from all the parameters, you're changing just one, which is the way it's counted. Everything else stays the same.
Exactly.
Nevertheless, there's a full array of competitors in that field, which go from, I think the fastest is Orb. They are marketing how they get to a result in 1 minute. And you're from this new wave, you're the slowest. Yeah. You're much faster than the incumbent, but you're slower than the others. So how do you position yourself compared to an Orb, which does it in 5 minutes?
I will ask one question. You have a dead body in front of you, okay? And you ask him a question. Do you think that he will answer?
I hope not.
Okay. So it's the same. All the technology that are able to detect microbes in a minute will never be able to differentiate the culturable microbes to the non-culturable microbes. So it will be always good to have this solution because you will have an alert, but you will not have the right number. And when you have an action that is linked to a threshold, you must obtain the right number. When you perform Legionella analysis and you obtain 10 to the 4 per liter, you have the obligation to take an action and to clean the pipe by disinfection. You need to recount by the standard method and see what will be the number. And the number must be, let's say, 0. If it's more than the threshold, you have an issue and you need to close definitively and clean everything. If you use a rapid method that is not able to make a difference between a culturable and a dead Legionella pneumophila, what will happen? You will make the disinfection, you will check your sample, the number of cells in the pipe will be always the same, but you will have only dead cells, and the dead cell will be counted by new fast technology as the same number. So the number will not change. So they cannot use it. And if you have a new technology, this online solution that count all the microbes, this new technology will not be able to detect the risk because they will not be able to count specifically the microbe of interest. I'm not saying that these new solutions are not good. They help to take a decision. And if you have a big mess, they will detect it. But if you have just a small variation of one specific microbe, it will be difficult for them to detect it.
Let me put that in my words,
please.
and then you tell me if I understood right. There are 3 main differences. The number one is, it's a bit like if you're doing gardening. If you're planting seeds, you know how many seeds you have, but before they start to grow, you don't know which one will grow. So if I'm planting 10 seeds and I'm counting the seeds, I know I have 10, but maybe only 7 will start to grow. And you're only waiting for the growth to start to say, okay, that one was alive. Whereas if you're counting at seed level, you have no clue to know if the seed is going to grow or not. So that's the first difference.
Exactly.
The second difference is that you are specific. Like if you're looking for one specific microorganism, then that is the specific one you're going to count and not the total of them if there are more than one. And the third one, which totally skipped my radar so far, so it's super interesting that you mentioned, is that if you have all of them which are alive and would be able to grow and everything, so in which case the result would be the same with all methodologies, but now you disinfect, and in the process of disinfecting, of course, you kill them, but you still measure the dead bodies floating around. You might think with another methodology that you have not done anything because you still have the exact same amount of bacteria inside. But if they are dead, you don't care that they are there. So you are able to measure the fact that only the alive ones are able to grow. So only the alive will be counted and not the dead.
Exactly.
So those would be like the 3 main differences. Yes. Now, that was my positive summary, but I'm still the devil's advocate. So I have one more devil's advocate for you, which is, if anyways, what I'm trying to do here is to have like my auto control, my approximate of the number, which will at the end of the day still be counted the official way with the Petri dish. How inaccurate can I still be while being good enough?
Let's say you have one bacteria per 100 milliliter, that if you drink this bacteria, you die. And there are no other choice. And if you know that these bacteria exist, will you be happy to use a rapid method that counts everything? Do you will take the risk?
What you're saying is that it's a risk management topic. If I'm counting something which is relatively insensitive, then maybe I can go with whatever is available. If what I'm counting is a high risk, then I cannot afford to take that risk of going imprecise.
This method that counts everything, if let's say you have 10 to the 3, 10 to the 3, 10 to the 3, every minute, every hour, you will say, okay, I have no issue because I have always the same number. But within this 10 to the 3, you may have one cell that is very dangerous and you will not be able to see it because it's on the background level, because you have always 10 to the 3. So what I am saying is all these solutions are good and I'm very happy that they exist, but it's always good to have also accurate and specific rapid method in parallel. It will not replace the rapid specific method. I'm not saying that all these companies are doing bullshit. It's not the case. They are very smart and we need them, but we are not in the same field because I'm working with laboratories and they connect directly to the pipe. It's not the same.
In terms of verticals and applications, would it be more of a drinking water topic, more of an industrial topic? Where do you see the best fit?
The best fit is where the value of the risk is higher. So you have two possibilities. If the product that you sell is very expensive, you will be happy to have very fast results and accurate to be secure during the production. So on pharma industry, cost is very important of the product. And if you have rapid method, you will be alerted very fast. This is one major application for us. And we have a worldwide partnership with one major company, I cannot disclose the name, but will release a product by end of the year using our technology for the bioburden, for total viable count. This is a first market that is very interesting. The second market is all the beverage market, because you have issue with the brand if you have a problem, and you have issue with the cost of what you lose when you destroy all the non-compliant product. So this is a second branch where we see a big opportunity. Unfortunately for us, and it's something that is important to have in mind, the water, the tap water costs nothing. And this is a big issue because it costs nothing. So why you will do rapid test?
Because you may want to protect the people, but
I agree with you, but...
You mentioned beverage. When you see a Perrier with all the trouble they are facing recently, those would be your obvious customers.
But I cannot disclose.
Of course.
But yes, we are discussing with a lot of large players like that in US, in Europe, and they are quite interested by our solution because they produce bottled water 24 hours a day, 7 days a week. If you produce 1 million bottles every day, and if you detect after 1 day that you have coliform present, you need to destroy 1 million bottles. So if you obtain a result not in 1 day with the standard method, but in 6 hours, you destroy 4 times less bottles.
That is exactly why I mentioned Perrier, because Perrier had this story of having to destroy 5 million bottles, and I was wondering 5 million bottles because of E. coli is very probably they have not counted in a fast manner. I have 2 topics which I really want to take with you still. One is your business model, because we covered how you're selling the Mica Advance box and also how you're selling the reagents needed to make every single test. I know that the Mica Advance, you don't have like so much backlog of data because it's pretty fresh that it's on the market. But in your vision, what is the share of CapEx sales and what is the share of recurring revenue? Because what you're describing sounds very much like an espresso.
Yes, it's an espresso model. And what we expect is in 2 or 3 years, we will have 75 or 80% coming from the recurring revenue and 20% coming from the equipment. And we are thinking even when we will have big player, that perform 300 analyses a day, the question will not be the equipment. We will give them for free the equipment. Because if you generate 500,000 euros per year of recurring revenue, why we will develop just by buying an equipment?
Which is the interesting question of where you set the bar of the type of growth you want to follow as a path. Because you mentioned how you need 2 million to develop a new parameter. One way might be to just sell what you have, wait for the recurring revenue to build your bank account. And once you have 2 million in the bank account, you empty the bank account and you add a parameter. That will provide you like a very de-risked, slow growth. That will very probably make Pure Terra, your investor, very angry at me for even suggesting the idea. No. The other extreme of that would be to say, you know what, let's take a big plunge and put the Mica box in every single laboratory for free, as you just hinted to. And then it's the Uber way. We're going to be a sinking ship for years. But the day that it starts to catch up and to go back to the surface, it's going to be like a rocket.
Exactly.
Because that day, the recurring revenue is huge. But between those 2 extremes, where do you set the bar?
It's a question of raising money. If we have enough money, we will be able to build more equipment and we will be able to install for free more equipment. So today we are discussing with some potential customer where we are saying, okay, we leave for you the equipment for free for 12 months. no problem. But you are engaged to buy this amount of tests. So we are working like that right now already. Because when you have a laboratory, maybe they have not planned the budget for the equipment and they will need 1 year or 6 months. And we cannot wait 6 months. We prefer to install the equipment today and they start work right away. We will not install for free all the equipment because we will not have enough money to do it. But yes, it's a strategy is to combine for free, partially for free equipment and to have as much as possible customer all around the world fast.
Let me put my French hat on. We are in France, the country of Pasteur, and you're in that field. You're in the legacy of Pasteur. You just in our conversation so far dropped how you've been in Senegal, in Macau, in China. I mean, you're making the French legacy of Pasteur shine. You should be a shiny object for French tech. You have a differentiated technology which is derived from something which obtained the Nobel Prize. So it's not like garage technology. It's like something which is universally recognized as something brilliant enough to get a Nobel Prize. Would it be totally stupid to think that you should get like massive support from all the French mechanisms which are in place?
We got all of them. We just obtained a grant from ADEME for the project of Mica Advance for the reused water. Okay. So we obtained just a green light for another grant, but it's not official. So I will not be able to disclose also for also the development of a new equipment that is in our pipe. And we obtained several times BPI, grant. So we have been really supported by the French authorities, but not by French laboratories. It's very funny. So it starts, we will normally install the 2 first equipment before end of the year with big laboratories. So we are quite happy.
So that means that Sanofi is not behind you yet?
No, not yet.
But they might.
Maybe. We will see. What I'm saying is we start to sell equipment in Macau, in Greece, in South Africa, in Dubai. But now we start to have some interest in France. So I was expecting the opposite, but it was not the case.
I'm wondering how you manage your go-to-market. Is it you hopping on a plane, going to a place and offering the solution, or do you team up with distribution channels?
The strategy since the beginning was to develop a network of distributors. So we communicate worldwide and we say, look, we have Mica, we have this technology, so maybe we'll be interested to promote the solution. And we got a very good, feedback from distributors. So when it's not close to France, we work with distributors. But working with distributors takes a lot of time and you need to train them. You need to explain them the way it works. And they have not only your product to sell, so you need to follow them. So at the beginning, I was alone to promote the sales of the solution. And in June 2024, we recruit the first salesperson. And in October, another one. So now we are 3 persons working together. And since that, we have doubled the number of distributors. So in just 12 months, which is quite good. But a distributor, it takes him between 1 and 2 years to learn the product before starting to sell it. So it's a long process. And because we make worldwide communication on our solution, we have been contacted by customers in US or in Trinidad, it was not expected, or in Tahiti or in Dubai or in South Africa, So it's also a question of communication. And because the issue of microbiological analysis is worldwide, you have customers that are looking for rapid solution. And when they have an issue, they contact us and we start to discuss and try to see if our product fits they need,
Which leads me to my closing question in that deep dive, which is your vision of the business. Because one way would be to say, okay, you keep hiring salespeople and developing your distribution channels. And as you said, you need to seed them. And then it takes 1-2 years until they go full steam and then they start covering their field and region. The other way would be to say, if you keep doing something which is irresistibly cool and demonstrates that you can go to that 80% of recurring revenue, companies will be lining at your door to acquire you. So do you want to build a giant, or do you want to be the future gem in Veralto's crown, to name one?
It's a good question. Maybe 5 years ago, if you ask me this question, I will say I want to be the new big company. But maybe now, after 10 years growing the company, and I say maybe being purchased by a company in 5 years or 3 years will be also cool. And I will do something else. I don't know yet. Today, what is important is to accelerate the sales, to grow the company and see the opportunity. It's always a question of money also, what it will be. And if I will be part of a big company and be able to accelerate all the development in terms of technology, maybe it will be good also. Because what we have done today is focusing our technology on water, but we could do exactly the same with human diagnostics, microbial infection. We could do exactly the same with antibiotic resistance. We could do a lot of things with the technology that we have developed. But because we have not a lot of money, we do only one thing. So if tomorrow we have been purchased by a big player and this big player understands the power of what we have done, maybe we will have enough money to develop and they will have to sell what we are doing, because I'm coming from science.
But I mean, you're in that phase where you're crowdfunding. So I guess that's music to the ears of your investors. Because if you're an investor, of course, you'd love the company to become a giant, but the best way to get your money out is for the company to get acquired. So I guess, yeah.
Exactly. I agree. But you could become a giant and make an IPO.
Yeah.
But
you could.
I will not recommend it, to be honest. Okay.
I was going to say that, but it's good that you said.
No, I will not recommend it. And I know what I am saying because I was on pre-IPO in 2019, So I know what's the mess and I know all the risks that are around it.
I'm glad for you didn't do it, because the track record for growth stage companies doing IPOs in water endeavors or water-related endeavors is unfortunately not that good.
So yes, the best strategy for me will be to be purchased by a company, but it's not the end.
So that's the vision, midterm vision. What needs to happen in the next 12 months so that you keep on track with that vision line?
So to be honest, we have some company that already knocked to the door.
Okay.
We have been contacted, and before end of June, I will be in one of these company installing equipment, and they will start to evaluate the technology in detail.
So due diligence even started. It's the right time to invest. It's not financial advice.
Which means that for an investing point of view, it might be the right time to invest, because when you start to have some big player that knock to the door and say, "Oh, I like what you are doing and I'm interested to evaluate the solution." Yes. And we have not contacted them, they just came to us.
I think that's, even for you personally, having pushed and grown the company for 10 years, it must be like a rewarding moment when you see big guys coming to knock at your door without you having to proactively go at them. I do know that for my very small little endeavor of my podcast, the thing which made me the most happy is when someone I didn't know and I had not contacted reached out to me and said, "I listened to your podcast and it's cool." I was like, okay, that means it's working. So I guess for you at a fully different scale and with fully different stakes, it must be something when a big player knocks at your door and say, "Hey, can I test it out?"
Yeah, I was happy. It was 4 years ago, when the first big player came to us and said, "We like what you are doing, your concept of microcolony, and we want to sign an exclusivity on pharma industry." And we developed for them the technology. And now we know that end of the year, the product will be released. So it was the first huge sign, a big player that performed more than 10 billion of revenue, So it's a huge one. So that say, okay, now we will put our brand of one of your technology and start to sell it on pharma. And now a second company came to us and say, "Wow, we like what you are doing and we are interested to evaluate you to acquire you." We will see. 12 months after, we will see.
Still not investment advice, but I started by saying that I was on the fence. I guess you convinced me. I'm gonna cut you a check, a very big check of right now, I can invest in you for the big sum of €242.33.
Right.
So I think that's within the type of risk I can take. So I'm going to take the plunge. But not investment advice, do your own research, usual stuff. I guess people had 1 hour of your explanations to make their ideas about that. Thanks a lot, Sam, for everything you explained. If that's fine with you, I have a last segment, which is the rapid fire question.
It's time for the rapid fire questions.
What is the toughest challenge, in your opinion, for a water tech startup?
sales cycle.
It's funny how often that one comes up. What would be your best single piece of advice for the founders and managers of the about 1,000 early stage water startups?
If you think that you will be able to reach the goal in 1 day, put 5 days.
What's the drop of knowledge you wish more investors knew about the water sector?
When I start to discuss, with investors on water, I understand that they know nothing at all. So I was a bit shocked.
What was your most unexpected partnership and what did it bring you?
It was my first partnership with the big player, and it was not expected because, when they start to knock to the door and say, "Oh, you know us?" Of course, I know you. They help us a lot because they explain us a way to develop a solution for a big group. So when we start, 4 years ago, we have developed the solution by ourselves. But based on what they are doing for the development of their own solution, we learn a lot.
Super short, profitability or growth?
Growth.
What is the next profile you'll hire?
A field engineer.
And when you hire that field engineer, do you look for sector experience or startup experience?
Sector, not startup.
Will you be opening new markets or doubling down on the current ones?
Not simple to answer.
What is that tool nobody speaks about, but you could not live without?
My computer.
What's the single piece of insight your ideal customer profile needs to hear right now?
Increase the productivity with our solution.
That's very clear. And honestly, that is one which I was also noting down during our conversation because I didn't realize how that plays a role. But that for sure is something which will make people click, I hope, for you at least. What are you desperately needing and want to raise an open call for right now?
I need money, It's crazy, When you see what we have done, we have raised in total 14 million in total, where some other company will raise 2 times this number and not reach where we are. So we're able to do very smart thing with very low amount of money. Of course, 14 million is a lot, but compared to when you see what we have done, you understand that it's not so much to do what we have done.
If I try to benchmark you, for sure, being a French or European company isn't the right side of the Atlantic to be on.
Yes.
But then necessity is the mother of creativity and invention. So you are able, being relatively bootstrapped, to do stuff which others don't do with much more money. You say 2, I would have said 5 or 10 times more. But last question, what can and should I do for you?
Connection, link.
That is within my real.
Yes. I like to meet people, and it's because we meet people that we have new idea.
Makes sense. Well, Sam, it's been a pleasure. Thanks a lot. I know that I went massively overboard, so thank you for not closing the call and saying, hey, don't be that French and don't be that long. So thanks a lot. I'm going to watch with great interest how your fundraising closes, and let's see what's next and who's that mysterious strategic which you're in talks with and in early due diligence.
Thank you.
Thanks for listening to Don't Waste Water. This podcast was brought to you by GF Piping Systems. Loved this episode? Head over to Apple Podcasts to subscribe, rate, and leave a review. See you next time.